Good morning, everyone. My co-moderator today is Tycho Heimbach. He's a senior principal scientist at Merck. And my name is Ethan Stier. I'm the Associate Director for Lifecycle Management and OCP at FDA. Both of us are really excited to be kicking this off. We have three excellent presentations by really world-class scientists. The theme will be using PBPK to assess the impact of food on absorption, and also focusing on using PBPK to assess bioequivalence under the fed state. The presenters that we have today, the first one, and I will try to introduce them briefly in between speakers, but I just want to announce them now. Dr. Cristofoletti, Assistant Professor from University of Florida, will be followed by Dr. Gousous, a lecturer from Johann Gutenberg, University Mainz. The other presenter for this presentation will be online today is Dr. Langguth, who's a professor also from the same university. The third presentation will be by Dr. Rebeka Jereb, Senior Scientist of Clinical Innovation at LEC. That presentation will also be given online. Rebeka will be joining us. And then I also want to acknowledge the panelists because when we kind of transition, Tycho and I want to make sure we have the maximum time for discussion because we really know that's why everyone is here in addition to hearing these great presentations. So that panel will include Dr. Ahmed, who's the VP and Head, Biopharmaceutics and Bioequivalence, Dr. Reddy's Laboratory, Dr. Susana Almeida, Clinical Development and Safety Officer, Medicines from Europe, Dr. Jianghong Fang, Senior Staff Fellow, DPM, FDA, Dr. Vidula Kolhatkar, Pharmacologist, DB2, Office of Bioequivalence, FDA, Paulo Paixão, who's a member of the MWP, as well as an assistant professor at the University of Lisbon, Mohan Rayeni, Scientist, Biopharmaceutics and IVIVC, Sandoz Development Center, and finally, Dr. Fang Wu, Senior Pharmacologist, DQMM at FDA, and Lei Zhang, Deputy Office Director, ORS, FDA. So again, let's give a warm welcome to Dr. Cristofoletti, who'll be giving the first presentation this morning.
Okay, thank you very much, Ethan, for the very nice introduction, and also thanks to the organizers for the very kind invitation to be part of this very nice meeting today here. So my talk today will be about an investigation of the usefulness of PBPK modeling to recapitulate formulation-specific food effect. So just a brief outline, I'd like to emphasize here that for this modeling exercise, we'll be focusing on two formulations containing itraconazole. We'll be focusing on Sporanox, for which we have a positive food effect, and also on Tosura. There is another amorphous solid dispersion, but for which we have a negative food effect. So our goal here was to investigate if we can recapitulate that with PBPK modeling. So just starting from the beginning, we dug the literature and we found clinical trial from the 80s with some PK data for itraconazole after intravenous infusion. But that data was obtained using itraconazole with cyclodextrin. So the first question that we got was whether we could use that data set to help us to develop the model, since our model formulations, they do not contain cyclodextrin. So just a very preliminary investigation. So in this PK plot, you can see that the upper PK profile is the profile for the IV administration of the formulation containing cyclodextrin, and the other PK profile is for a formulation without cyclodextrin. So as you can see there, we can observe a parallel decay in itraconazole concentrations from both formulations. So it was kind of reasonable to assume that the cyclodextrin in the IV formulation was not affecting the distribution, so we could indeed use that formulation to help us with the model development. So the first analysis that we carried out for this IV data was a compartmental analysis, and the data set was best described by a three-compartment model. And what was very interesting was that the K31, that is the redistribution rate from the peripheral compartment back to the central compartment was the slowest step in the distribution of itraconazole. So that was more like the signature profile for itraconazole after IV administration. And then we moved on towards developing the disposition model, the disposition PBPK model. So using that IV data set. So we collected some additional drug-specific parameters from the literature, blood to plasma ratio, fraction unbound, log P, and we decided to use the Rogers and Rowland method to estimate the KP values and the VSS. So, and I mean, what happened for itraconazole, it's not only for itraconazole, we noticed that for other highly lipophilic weak bases, is that normally this Rogers and Rowland method, it tends to overestimate VSS. And that was pretty much the case for itraconazole as well. So in that case, it was really necessary to apply an empirical scaling factor to recapitulate the clinically observed volume of distribution. So in our case, it was necessary to use a KP scalar of 0.2. So just getting back to Dr. Jusco's presentation at the beginning, I think we have a gold opportunity here to try to revisit some of those tissue composition-based KP algorithms to investigate why they are generally overestimating the distribution of highly lipophilic weak bases. But here in this plot, we have a very preliminary simulation of the distribution of itraconazole in a full PBPK model. So here basically we have the PK profile in different compartments of the PBPK model. And interesting, we can also see a kind of a triphasic pattern over there. So we can see that for the drug distribution into the muscle compartment, we can have a very fast distribution into the muscle compartment, achieving the higher concentration. For many other compartments, we can see that the profiles, they are just overlapping. And what is quite interesting is the distribution into the adipose tissue. So we can see there in the plot that we really have a very slow redistribution of itraconazole back from the adipose tissue back into the systemic circulation. So the PBPK model, even though it's quite preliminary at this point, it was also reproducing this triphasic behavior that was observed in the compartmental analysis. So that was a good sign for the model development. So in terms of elimination, we just applied the retrograde model in SynSIP that is really nothing but back calculating the intrinsic liver clearance from the IV data. And after doing this, after back calculating this intrinsic liver clearance, we were able to scale that down towards the abundance of the enzyme. So we were able to estimate this intrinsic clearance related to the abundance of the enzyme. And with that information, and also with some unbound in vitro Km values that was published by folks from the University of Washington, we were able to back calculate the Vmax value for itraconazole as well. So that was pretty much all the inputs for the disposition model for itraconazole. And here we have a comparison between the simulated and observed clinical PK profiles for itraconazole after an IV administration. So the green line represents the simulation and the dots, they represent the clinical trial, the clinical results. So there is a very good agreement between the simulation and the clinical observations. And we can see the comparison, the ratio between the simulated and observed AUC 0 infinity was 0.9, approaching the unit. So that was that gave us enough confidence to move on with this disposition model for itraconazole. So we were very fortunate to find a very rich data set for itraconazole. So we were also able to interrogate the external validity of this model. And here you can see that the model was able to recapitulate also multiple doses of this IV infusion. So once again, the lines represent simulations and the dots, they represent clinical data. We have two simulations in this plot. The simulation represented by the dotted line is a simulation where we are not considering a potential autoinhibition of itraconazole. So we can see that without considering the autoinhibition, we are underestimating the PK profiles, and the solid line represents the same simulation, but now considering a Ki value for itraconazole and also for its metabolite. So when considering the autoinhibition of CYP3A4, we were able to really recapitulate the PK profile after multiple administrations. So the model was able to capture this time-dependent change in clearance. So we were able to recapitulate the clearance after the first dose and also at the steady state. So we were pretty satisfied with the disposition model at this point. So it was time to incorporate the absorption part of the PBPK model. And here we decided to apply a kind of a stepwise model-based approach. So focusing on analyzing in vitro data individually to estimate fundamental parameters that would be used as inputs for the PBPK model. So basically we analyzed each data set on a separate way, starting with solubility, then dissolution, and then precipitation data set. So for solubility, the first question that we got was whether it would be useful to try to parameterize the model using equilibrium solubility for the crystalline form of itraconazole. We have done that. The data is not here, but we just got a massive under-prediction of the PK profile. That was really not explaining the PK profile for the amorphous solid dispersion. So when we were investigating potential solutions for that, we ran into this very interesting article from which I got the table on the left-hand side of my slide here. So basically, they analyzed the solubility of the different solid dispersions with itraconazole. The important one for us is the one with HPMC, is a solid dispersion with hypromelose. This is pretty much sporanox. And if we compare the solubility of the solid dispersion with the solubility of the crystalline form, we can see that the solubility of the solid dispersion is almost 100-fold higher than the solubility of the crystalline. And this solubility experiment was done after 24 hours of incubation. So it really seems that we can achieve a very high level of supersaturation, and this level of supersaturation for the solid dispersion is stable over a physiologically relevant time span. So because of that, we decided to investigate to quantify the solubility of the ASD. So basically, we just used the content of sporanox capsule for solubility studies. And the results are on the plot on the right-hand side of my slide. In orange, we have the solubility of the ASD, and in blue, we have the solubility of the crystalline form under the same experimental conditions. So for all the solubility measurements that we have there, not all of them are equilibrium solubility for the ASD, but at least the values that we got for apparent solubility, they were stable for at least 2 to 3 hours. And we assumed that was good enough from a physiological perspective, and we decided to use those solubility values for the ASD to inform the PBPK model. In terms of dissolution, that was a big discussion here. So one of the goals of this modeling exercise was to investigate sources of the between subject variability in the PK of itraconazole. So applying the conventional approach related to dissolution, and by that I mean just direct inputting the dissolution profile into the PBPK software was not going to work for us, because when we do that, we are kind of bypassing all the GI variability in the virtual cohort, and we are telling the software that the dissolution is going to be the same in all virtual subjects, regardless of the composition of the GI tract. So we decided here to apply more like an iterative process, combining the convolution of in vitro data to estimate fundamental parameters and reconvolution of these parameters back to the process, alternating between SIVA and SynSIP. So basically, in SynSIP, we have this diffusion layer model. The equation is represented on the slide. And we have, in this model, we have the solubility, we have particle size, and we have an empirical scaling factor. So here, we decided to fit this model to in vitro data to estimate a potential apparent particle size that this is going to be a surrogate that together with solubility data would enable us to reproduce the in vitro dissolution of sporanox. The problem with this type of approach is that we may struggle with identifiability issues. So in order to mitigate these identifiability issues, we fit this model to different experimental conditions, different experimental data sets. So basically in this exercise, we fit the diffusion layer model to 3 different data sets in simulated gastric fluid, FASGIV, and phosphate buffer, USP phosphate buffer. So in here, we don't have only differences in terms of pH and composition of the media, but we also have differences in terms of the rotational speed. So, and after fitting the diffusion layer model to all three data sets, we were able to estimate this particle size of around 9.7 micron, with a very decent precision around it, with an RSE of only 28%. So here, in order to investigate the robustness of this particle size, we ran a real prediction. We simulated how would be the dissolution of this Sporanox formulation in Tausif. And we got our simulation, and then we went back to the lab and ran the in vitro study. And as you can see in the plots over there, we have a very good agreement between the simulations, all the lines, and the in vitro data for all experimental conditions. So it seems that this particle size that we fit to in vitro data was really able to explain the dissolution of Sporanox across different experimental conditions. So that gave us enough confidence to use this as an input for the PBPK model. When it comes to precipitation, I think we were very fortunate here to have access to data from a two-stage biphasic dissolution study. So this is more like, this is offering us an open system to investigate the precipitation of Sporanox. And here, we simply fit a biphasic dissolution method that is available in DDD+, to simultaneously to the concentration of itraconazole in the aqueous and organic phase. And with that, we were able to estimate a precipitation rate constant of 1.3. So our parameters for the PBPK model. So we got our precipitation rate constant, a parameter describing the dissolution of Sporanox and solubility of the SCD. We used all of that to inform the PBPK simulation. And here we have our first result. So every time that I'm showing this, the lines, they represent the simulations. The green line is the mean value, and the dots, they represent the clinical trial. So here, it was necessary to further optimize the precipitation rate constant. So it was a fitting exercise, but I would call your attention to the table on the bottom part of my slide. So even though there was this fitting for the precipitation rate constant to further optimize this precipitation rate, the model was able to recapitulate the between subject variability around the PK parameters. So we have the simulated CV%, this is the between subject variability, and the observed CV% for the two PK parameters. So we can really see that for Sporanox, we have a very high between subject variability, and the model was kind of recapitulating that, the same level of variability as well. So, but since we got this fitting exercise, it was still necessary to interrogate the external validity of the model. And here we have another simulation using the same model, but right now, simulating the administration of 200 milligrams of Sporanox, meaning two capsules. So once again, there is a very good agreement between simulated and observed PK profiles in terms of central tendency, but also in terms of recapitulating the between subject variability. We got another opportunity to externally validate this. So basically the same model again, but right now we apply this model to reproduce a clinical trial that was carried out in healthy volunteers pre-treated with ranitidine. In this clinical trial, they measured what was the gastric pH after pre-treating the volunteers with ranitidine, and the median value was 5. So basically, we applied exactly the same model that was used in the slide, in the previous slide, just changing the gastric pH, representing this hypochlorhydric situation. And once again, the model was able to recapitulate the clinical trial. So we got enough confidence in terms of this Sporanox PBPK model under fasting conditions. And it was time to switch gears towards the other formulation, that is Tausura. So as I mentioned, both are ASDs. In Sporanox, we have itraconazole with hypromelose. Hypromelose is a pH-independent polymer. And in Tausura, we have Sporanox with hypromelose phthalate. That is a pH-dependent polymer. So Tausura, meaning that since it has hypromelose phthalate, it only releases itraconazole when the pH is above 5. So, and Tausura is not bioequivalent to Sporanox. So here we have the result of a bioequivalence study for comparing Tausura 65 milligrams with Sporanox 100 milligrams. So even though the the dose of Tausura is smaller than the dose of Sporanox, the systemic exposure generated by Tausura is much higher, is higher in comparison to Sporanox. So they were not bioequivalent, and we can see that the confidence intervals, they are exceeding the upper limit of the bioequivalence criteria under fasting conditions. And on the bottom part of the slide, we have the in vitro results comparing both formulations, in orange Tausura and in blue Sporanox. So since we have Tausura with this pH-dependent polymer, we don't have a significant dissolution of Tausura under simulated gastric conditions. But things are completely different in simulated intestinal fluid. So for Tausura, we have a very fast dissolution, achieving 100% in less than 10 minutes, and this supersaturation level is stable for at least 2 hours. On the other hand, for Sporanox, we have a very slow dissolution. So we repeated all the steps related to analyzing in vitro data for Tausura as I described for Sporanox. Just for the sake of time, I'm going to skip that. And here we ran a virtual bioequivalence simulation. But here we are not propagating the within subject variability. We are just comparing the point estimate, the geometric mean ratio, simulated geometric mean ratio with the observed geometric mean ratios when comparing Tausura and Sporanox under fasting condition. So, as you can see, the simulated GMR for AUC was around 1.21. The clinical observation was 1.23. So for Cmax, we got a simulated GMR of 1.6, really indicating that they are not bioequivalent because the point estimate is completely outside the bioequivalence criteria, and the clinical trial got a confidence interval of 1.4 to 1.85 with a GMR of 1.62. So it seems that the PBPK models under fasting conditions, they were really recapitulating the performance of those two different formulations. So I'm just running a little bit over my time here. So right now it was time to apply the models to investigate the food effect. So in order to investigate the food effect, we used a novel biorelevant media, the name is fed gas, that was developed really to emulate the stomach content after oral intake, after intake of FDA high-fat breakfast. So this is the composition of this fed gas, and it's a gel that we can mix with water. And for the fed gas, we have 3 specific conditions. We have fed gas early, fed gas mid, and fed gas late, simulating the dynamic, or better, the time-dependent pH in fed stomach. And here we were able to run in vitro dissolution studies for Tausura and Sporanox using this fed gas. Let's start with Tausura. We can see that the dissolution of Tausura in fed gas early, when the pH is 6, is very fast. So we are achieving 60% dissolved in 1 hour. But when we change the pH of the media to represent different stages of the fed stomach, the dissolution decreases. And the complete opposite behavior is observed for Sporanox. So basically, we are just using this new in vitro information as the inputs for our PBPK model. And here we have a simulation showing Sporanox under fed conditions. And once again, the model is the same PBPK model right now, just with a new input related to dissolution in fed gas and FASIF. We were able to recapitulate the PK profile of Sporanox 100 milligrams under fed conditions. And once again, the model was able to recapitulate the between subject variability. The variability is smaller under fed conditions in comparison to the fasting condition. The same simulation for 200 mg, and the result was again positive, so the model was able to recapitulate the central tendency, but also the viability around AUC and Cmax. And for Tausura, since we have a pH-dependent polymer, our hypothesis here was more like, we have a very fast dissolution of Tausura at the early stage, right after administering a meal. But since the pH of the stomach is changing over time, when this pH is decreasing, we may have precipitation of Tausura because the polymer is not going to be soluble when the pH is below 5. And we ran a 3-stage dissolution in fed gas, transitioning from fed gas early to fed gas late. So you can see in this plot, we started the dissolution study in fed gas early, pH 6. The dissolution of Tausura is quite positive and fast. And then we added a bolus of hydrochlorhydric acid to push this pH back to 4.5, representing the fed gas mid. And we can see that there was a precipitation of the dissolved Tausura there. And then again, sometime after that, there was another bolus to represent this fed gas late, pushing the pH to 3, and there was another precipitation. So the hypothesis here was more like, we don't have just one solid state in the system anymore. We have two solid states. At the beginning, only ASD, but when we are changing the pH, we are acidifying the conditions, the polymer is precipitating, and that is kind of crashing the supersaturation, crashing the solid dispersion, and we have the precipitation of itraconazole as the crystalline form as well. And that was the input for the PBPK model of Tausura. And when considering that, we were able to recapitulate the slightly negative food effect on Tausura pharmacokinetics. And here we have a comparison between predicted and observed clinical data for Tausura 65 milligrams under fed conditions. So meaning that the model was able to recapitulate the PK profiles under fasting conditions and also the PK profiles under fed conditions, accounting for the different impact of food on different formulations. So I'd like to thank all the collaborators for this, Dr. Nitin Sharp in my lab, Professor Jennifer Dresman and Niklas Rudolph from Fraunhofer Institute, and Professor Jim Pauly and Roshni Patel, Ana Luisa Coutinho from University of Maryland, and also to the FDA for sponsoring this research. Thank you very much. Looking forward to having fruitful discussions.
So thank you. Great presentation, Dr. Cristofoletti. So now we'd like to welcome Dr. Al-Gousous from Johann Gutenberg University Mainz, and his co-presenter will be online with us today, Dr. Langguth.
So now it's afternoon. Good afternoon, everybody. So today I'm going to present some of the work we are doing regarding dissolution and disintegration in the fed state. So now one of the aspects of dosage form of oral solid dosage form performance in the fed state is the effect of the increased viscosity. Now when food is taken in, the viscosity of the gastric fluids increases, and this has effect on the drug release from the dosage forms. So the dosage form disintegration and dissolution will be slowed down. Why? Because higher viscosity would retard water penetration into the tablet according to the well-known washboard equation, where, as you see, the rate of penetration of fluid into the tablet core would be inversely proportional to the square root of the viscosity. And based on that, disintegration and accordingly dissolution can become 1 to 2 orders of magnitude slower. And when characterizing this in vitro, we have got a problem that USP type 1 and 2 dissolution apparatuses are not that suitable. Why? Because the high viscosity, it results in the flow becoming laminar. The Reynolds number will become low, and low Reynolds number means elimination of eddies, and eddies are the major uplift mechanism for particles in any stirred tank, like a dissolution vessel. So we will have very strong coning effects. To overcome this challenge when characterizing dissolution in vitro in viscous media, we developed this apparatus, computerized numerical control, CNC operated, which we introduced first introduced in a publication in 2015, but now we are developing it further. We have this basket mesh assembly for 3 tablets, and within a 1.5 liter tank containing the viscous medium, which our viscous medium of choice is for now 1.4% HPMC, it can move up, down to the sides and have a circular motion. So this motion patterns in all directions in 3D greatly reduces the cone formation. And here we have a case study with this apparatus using midodrine HCl. Midodrine is a prodrug used for orthostatic hypotension, and we have publicly available PK data from an ANDA for 2 IR tablet products. So first of all, we started with modeling intravenous and solution data from an old publication from 1987. And we made what we can consider a semi-physiological PK model. Semi-physiological to the effect that the processes that are before the drug crossing the intestinal epithelium are modeled based on physiology. Like we have gastric emptying, we have also the degradation in the lumen, and we have the small bowel transit time into the colon. So we physiologized the pre-absorptive processes. As for the post-absorptive processes, we kept them to be modeled using classical compartmental modeling. And this physiologically adjusted compartmental modeling provided very good fits for the data, both for the parent and for the metabolite. For the metabolite, data are not shown. So we have, after we have succeeded in modeling the oral solution, we can go and incorporate also a dissolution step based on that model. So we added a dissolution step in the stomach in the fed state. Now we assumed that here we made an assumption that disintegration would be the rate controlling step of dissolution. So gastric emptying occurs only for dissolved drug. So, and accordingly, dissolution would occur only in the stomach. Now, so we added a dissolution step in the stomach prior to gastric emptying. Now, the dissolution step, we modeled it using a diphasic dissolution model that we introduced before in one of our publications in 2020. Now, because now the dissolution process in the stomach under such conditions would be very complex, because you have shifting particle size, you have shifting dissolution media properties like pH and buffer capacity, and you have shifting viscosity. So to fit it, it would be maybe better to, instead of using kind of a simple viable model, to have something a bit more complex. So we use a diphasic dissolution model where the dissolution is supposed to occur in 2 phases, early and late, with a first order rate transfer constant between the early and late phases. And here are the results of the fits. This is the myelin product for which there are publicly available bioequivalence data. And as you see, the fits look quite good. Now, the remarkable observations would be the gastric emptying. I mean, the gastric emptying is slowed down compared to what we have typically in fasted state. Like in fasted state, the gastric emptying half-life will be roughly 13 minutes. Here it is more, it is roughly 22 minutes maybe. So the gastric emptying was slowed down, but not as dramatically as one would expect. This actually supports the hypothesis of the existence of Magenstrasse, that the mixing of the liquid that has been drunk with the chyme contents is not complete. So there is some separation between them, so a large portion is emptied relatively rapidly through the Magenstrasse. Other observation is that the first order rate constant for the luminal degradation was almost annihilated, it almost disappeared. I mean, there is a complete inhibition for the luminal intestinal degradation, probably because of the competition by dipeptides coming from the digestion of food. As for the Ka, I mean, there could be a small increase, but it is not substantial. So the competition effect on the dipeptide transporters does not seem to have that strong effect. And the post-absorptive pre-systemic metabolism does not seem to be strongly affected. Now, when we used this model to calculate in vivo dissolution for the two products involved in the ANDA, which is the myelin product, the test product for which we also have in vitro dissolution data, and the proamatine product, the reference product, which is no longer on the market, but there is one product which looks similar to it. So those are the dissolution profiles. So yes, the dissolution is still not fast despite the existence of Magenstrasse. So this indicates that the fluid in this Magenstrasse, okay, it is not completely mixed with the highly viscous chyme in the stomach. However, it is also not as thin as water. There is some degree of mixing. There are some viscosity enhancing components from the food that quickly pass into it. So it is not as thin as water. It is not as thick as completely blended FDA meal, but it is also not as thin as water. Now, in the late phase, we have very slow dissolution, which is probably tied to some of the drug particles being entrained inside the highly viscous chyme portion of the gastric contents. And there is one surprising observation that proamatine, which does not contain a super disintegrant, releases faster, a little bit faster. I mean, the two are similar, but still there is some faster release from proamatine, also indicated by the higher Cmax in the profiles, despite the lack of super disintegrant. So this, as we later discussed, could be related to mechanical forces. Now, when we make in vivo versus in vitro comparison, now this is the in vivo profile for myelin, and this is the in vitro profile in our CNC apparatus using 1.4% HPMC. 1.4% HPMC, we did it based on the rheogram comparison between the different HPMC solutions and fully blended homogenized FDA meal. So, therefore, there was a degree of underestimation of the dissolution rate, because apparently much of the dissolution occurs in the less viscous Magenstrasse, except for a slow final portion. So after further discussions involving also our colleagues in the FDA, we tried out the 1% HPMC solution. So you see in the figure above, we have the proamatine and myelin in 1.4% HPMC compared to the solid curves, which are the in vivo ones, and there is strong underestimation. And down we have 1% HPMC, where the curves, I mean, are predicted clearly better in 1% HPMC. This has probably to do because, I mean, the viscosity, okay, the HPMC meal, first of all, the fully blending HPMC meal does not account for the incoming gastric acid from the parietal cells, which dilutes it and decreases the viscosity, and on the other, and also it does not account for Magenstrasse. So 1% HPMC actually might be a better choice. And I mean, the curves overlap strongly, but we still have one problem, that the curves are predicted to be close to each other, but still they are not still sufficiently close to each other, and the rank order is still reversed. So this indicates, okay, there is another factor in addition to viscosity at play, which is probably what? I mean, for a highly soluble drug, it is probably not related to surfactant, probably related to surfactants or something. So our hypothesis is the mechanical crushing force, because in the fed state, the peristaltic contractions of the stomach get stronger. So there is a mechanical crushing force, and maybe one of the formulations is more sensitive to the mechanical crushing force. I mean, maybe the apotex or proamatine. Now, as I mentioned, the proamatine was discontinued, but the apotex formulation is similar to it. So, and we are also ask our colleagues in the FDA to investigate to which degree it is similar if they can. So maybe it is more sensitive to the mechanical force. And we have a hypothesis about that, but it is still too speculative, so I will not be mentioning it. So to incorporate mechanical force, for cases where mechanical force would be important, for formulations where mechanical force would be important, we started with another dissolution device design, which is based on a flow-through concept. So we have this device with the medium flowing around the tablet, like a large flow-through cell, and the tablet is subjected to mechanical stress using a force probe. And this force probe, in addition to subjecting the tablet to mechanical stress, can also record the decrease of the tablet thickness with time. So we can get both dissolution and disintegration profiles. So here we have some initial results with different applied forces, different those profiles that are decreasing, those are the disintegration profiles that show you how the thickness of the tablet decreases with time, and the rising profiles are the dissolution ones. So here we can monitor concurrently dissolution and disintegration. And one thing that one insight that this already provides, I mean, these are still very initial data, we are only at the start, in the coming months we will have more, is that really under those viscous conditions, disintegration seems to be really the rate limiting step for the dissolution under fed conditions for highly soluble, for IR formulations of highly soluble compounds. So in conclusion, we can say that the fluid within the Magenstrasse is viscous, but not as viscous as a completely homogenized meal, which is an important consideration to take, because there has been some discussion about that, for example, if the dosage form disintegrates quickly, then it could behave like in the fasted state. Okay, it will be close to the fasted state, but not quite like the fasted state. To be very similar to fasted state, it needs to disintegrate extremely quickly in vivo, not just quickly. And complete dissolution within the Magenstrasse is often not likely. I mean, it is not, because it is not as thin as water, it is not that common that the dosage form dissolves like super quickly. There are some, but they have some special properties for that. And the need to incorporate viscosity into dissolution testers create challenges, like the problem with the uplift in the traditional USP devices, which needs to be tackled, and it could be that mechanical forces also play important role, at least for some formulations. So at the end, I would like to acknowledge all our entire team, Professor Langguth, Dr. Thomas Navrot, our PhD students, Valentin Stahl and Farouk Omer, and also our FDA colleagues, some of whom are already here, are now present with us now. I mean, I recall, I have seen already Dr. Fang and Dr. Abdullah Shuaib, and perhaps others are here still also, whom I have not noticed yet. So thank you very much.
Great, thank you so much for the presentation. So our last speaker today, before we get into the panel, will be Dr. Rebeka Jereb, and she'll be joining us online. So the presentation's coming up, okay.
So first, something about the effect of food, then continuing with bioequivalent studies under fed conditions, a case study I have to present, and then some challenges and opportunities we see as a generic company. With regards to the effect of food, I'm sure you're all well aware of it. So the changes it causes in the gastrointestinal tract, the changes in the transit times, the fluid volume, the pH, the parasites, and so on. In addition, possible binding to food components, chelation, differences in viscosity as mentioned in the previous presentation, and so on and so on. So all these changes might impact the drug dissolution and absorption, resulting in positive, negative, or no food effect. With regards to the bioequivalence studies, and coming back to the ICHM 13A, which was mentioned also in previous presentations, it describes the bioequivalence study that should be conducted for orally administered IR products. And in principle, single-dose studies under fasting conditions provide greater discrimination between the PK profiles of the test and reference and are the preferred ones. But the selection of fasting and fed bioequivalence studies depends on the comparator product dosing instructions, safety-related aspects, and the classification of products as high or non-high risk products. And basically, food can have differential formulation-dependent impact on the absorption of drug substances from high-risk products. That means for high-risk products, formulation design and manufacturing process are important because the in vivo performance will be impacted differently by varying gastrointestinal conditions between the fasted and fed states. And for high-risk products, fasting and fed bioequivalence studies are required. Below are just some examples of those formulations. But now looking at this thing from another perspective, for new drug applications, the current regulatory guidelines recommend an early assessment of food effect as well as pivotal food effect study on the to-be-marketed formulation. And Kesisoglu and colleagues recommend that based on their extensive analysis of food effect studies performed by the originators, they said that the repeated food effect studies should actually be approached on a case-by-case basis, taking into account the totality of evidence, including the use of PBPK modeling. So this includes the cases where there are formulation or process differences between the early formulation and final to-be-marketed formulation, because the repetition of food effect study did not give any additional information on those cases. So this would somehow lead us to the conclusion that food effect does not necessarily change with formulation or manufacturing differences. And then taking into account the totality of evidence, including the PBPK modeling, there comes this question: can fed bioequivalence study be weighed for high-risk products in two cases? The first one is where the test formulation has similar composition and manufacturing process compared to the RLD, and the second one when there are differences in the composition and manufacturing process compared to the RLD. Just an additional information supporting our thinking was our in-house analysis of 125 bioequivalence studies with just poorly soluble APIs. That was published by my colleague Diane. And he reported that no association was found between fasting and fed conditions and bioequivalence outcome. That means that fasting and fed studies were failing at similar rates. And there was no case where results under fed condition would be more discriminatory than the results under fasting conditions. So there was no case where a study under fed conditions would be bioequivalent and the study under fasting conditions would be non-bioequivalent for the same molecule. So this again leads us more to the thinking of the necessity of fed bioequivalence study. Now coming to my case study, for this drug product, fed bioequivalence study was required per product-specific guidance. This was a BCS class 2 drug with low solubility above pH 4. Pilot bioequivalence studies in fasting and fed conditions were conducted with 2 test products and 1 reference, and both test products were bioequivalent to the reference. Then some changes were made in the test formulation from pilot to pivotal study. And then the pivotal bioequivalence study in fasting conditions was performed, and it showed bioequivalence to the RLD. And then we have this question: can the pivotal fed bioequivalence study be waived? We developed the PBPK model in GastroPlus. We followed the good practices. So using the IV literature data to inform the compartmental disposition model. Because there was no available permeability data, we optimized it. We also optimized volume of fluid using the RLD data from pilot study under fed conditions, and using appropriate dissolution method with FASIF medium, the IV-IVC was developed. This approach was presented to the health authority, and they found the approach reasonable, having some comments, so saying that the model should be adequately validated and acceptable, and that we should provide additional data such as dissolution at multiple pH in biorelevant media, formulation composition comparison. And they also emphasized some model deficiencies, such as lack of validation. We developed our model only for simulating and predicting fed conditions. So they suggested that we should use also data from fasting, pilot, and pivotal studies, and also to use data from different doses or batches with different release rates. So preferably we would use non-bioequivalent batch, which we did not have. They also suggested that we should additionally justify the low fluid volume, which was optimized during the model development. This approach was not pursued further because bioequivalent study in fed state was conducted, and it was successful, bioequivalent, and in accordance with our predictions. So that's where this case study stops. So it was not regulatory accepted to waive fed B study in this case. Now, coming to challenges we have with PBPK modeling, just in general, when starting to develop any model, we face the non-availability or even contradictory values for different model parameters, such as the solubility or the solubility of amorphous formulations. So should we use API solubility? Should we use intermediate solubility, as was also mentioned in previous presentations? We also have difficulties with implementation into the model of different dissolution, especially of two-stage and the precipitation if it's happening. Sometimes we don't have any values for some parameters and optimization has to be performed. And this is even more complex where there are more parameters, and then this simultaneous optimization is problematic. There are still not clear things such as physiological parameters that are used in the even the commercial softwares, especially with regards to the variability between subject variability and within subject variability in the gastrointestinal tract parameters. Now focusing more on the food effect models and problems, challenges we have there. So the changes in the gastrointestinal tract times, transit times, those can be easily incorporated into the model. The changes in time with regards to the food effect on pH and fluid volume, those could be incorporated, but it's even more complex to do that. But some sticks are more difficult to implement, such as the impact of increased viscosity. Is it only impacting the transit time, or are there any other impacts, and how to incorporate those into the PBPK model? There is also this possible binding of the API to food components, or chelation happening, or degradation. So all of those are possible to implement, but you have to find a good input value or in vitro tests, and then to input those value into your model. Also, some other problems with food effects models are how to incorporate the impact on influx and efflux transporters or enzymes if food has those effects, the impact on lymphatic uptake if there are any changes, the changes in the permeability, and especially difficult it is to find to biopredictive in vitro method for negative food effect. But to conclude on more bright side, what are the opportunities we have? So the originator usually conducts many studies and publish results in articles or submission files, and those could be used for model validation. But on the other hand, we do not have all the relevant data to really use those data for model validation because, for instance, differences between formulations tested in different studies, we don't have all the details. And also interstudy differences, so being conducted on different populations by different CROs, all those things might lead to higher prediction errors. Also, on the sponsor side, more bioequivalence studies are usually conducted for different scopes, and we have pilot and pivotal studies in fasting and fed states, and all of those could be used for model validation. But again, these inter-study differences, studies being conducted in different populations, might lead to higher prediction errors, and this leads the sponsor to thinking, should different models be developed or fitted for each scope? So one for US, one for EU, one for Japan, and so on. Now, some best practices we try to follow, as mentioned before, with regards to the solubility of amorphous drug. Sometimes we use the solubility of the intermediate or final formulation to improve the model. With regards to the passive and passive biorelevant solubilities, there are different proposals for different media composition if the usual ones don't work. The probability, we usually try getting data from in vitro, any published data, or fitting using oral solution. And as I mentioned before, for disposition parameters, of course, we also try to find the IV data, but for some low soluble drugs, there just isn't any. So then we have to follow some other paths. Yeah, with regards to the validation of non-bioequivalent batch, we all know that that is what regulatory want to see. But sometimes it is just not possible to manufacture the batch that would be non-bioequivalent. We see that one possibility could be using the hypothetical batches, somehow simulated dissolution profiles, for example, using DDD+. With regards to virtual trials, for the number of subjects, we use the same as in vivo studies, which should be properly justified. With regards to the number of simulated studies, that is one thing I would say is still not agreed upon. Usually the regulatory comment is that it should be properly justified. And with regards to the variability, this was also presented in previous presentations, that it is best if you match the in vivo and in silico inter and intra-subject variability in PK parameters. And this is my last slide. The best practices related to food. As I said before, if you have a drug substance with pH-dependent solubility, the model should account for changes in pH with time. And with regards to the biopredictive dissolution methods, those should be developed as recommended from the health authorities for both fasting and fed states. But my question here, or my thinking was, how is validation in fasting conditions? So having discriminatory method in fasting really, really relevant for predictions in fed state if different method is used. So we are actually more focused on predicting fed bioequivalence and having the discriminatory method for fed conditions. Why do we also have to establish this discriminatory method in the fasting conditions? I believe, I understand that we gain more confidence overall in the model, but is it really necessary? Thank you.
Thank you, Dr. Jereb, for that presentation. So at this time, I'd like to welcome all the panelists up to the stage, also including our presenters that are here. And of course, we'll be having some of our panelists join us virtually today. And just because we are pressed for time, I introduced the speakers and panelists earlier. So for time purposes, I won't reintroduce, but I please refer you to the biographies that we have on the website to find out more information about our panelists. Okay, we're gonna just dive right in. Also, my co-moderator Tycho will help, time permitting, if we have some online questions from our online audience. But we'll first start off with our questions that we've established before. So like to kick this off, the first question will be going to Dr. Jereb online. If the purpose of PBPK modeling is to waive fed bioequivalent studies, is additional validation under fasting conditions necessary?
Yes, that was the question I ended my presentation with. So I actually repeating myself, but I understand that it is best if the model is validated in both fasting and fed conditions in order to get additional confidence that all mechanistic processes are properly captured, including the food effect. However, I would say that if a model is validated with only one batch in fasting state, and then the food effect is properly captured and the fed state is properly validated with different batches, so having different in vitro and in vivo behavior, that is at least 10% differences between batches, I would say there is no need to really seek for additional in vitro method to have this validation of different batches also in fasting conditions. As we are focusing on the prediction of the fed bioequivalence in this case. Thank you.
Great, thank you. We'll now hear from Mohan Rayeni, also online.
Yeah, thank you for the question. We do have the same experience as Rebeka has mentioned, but however, agency believes in totality of evidence, and it includes like proving the validation through non-bioequivalence batches, food effects, and by lower strengths.
Great, thank you. Now we'll ask the question also to Dr. Fang Wu.
Yeah, thanks for the question. Currently, I think our thinking is that using a fasting conditioned PK to validate the model will increase our confidence on the model, that the model can capture the food impact on drug exposure. But we also understand the challenges for predicting the food effect. So our consideration is that probably you get better understanding of the excipient and the food interaction using in vitro testing and how that can impact differently on reference and test product. That would be helpful. Yeah, that is, and also if you have the pilot study and the in vitro data, and you can demonstrate the biodiscriminating of the PBPK model and the fat condition, that will also increase our confidence.
Great, thank you, thank you. We'll move on to question 2. How can we generate a biopredictive dissolution to be incorporated into a PBPK model for predicting the PK under fed conditions? I will start off with you, Jozef.
Okay. Well, I think the most important consideration we need to take when dealing with IR dosage forms in this regard, let us restrict the discussion, maybe focus on the tablets for now, would be whether the compound is highly soluble or not. Now, if we are dealing with highly soluble BCS class 1 or 3 compound, then the main issues we need to focus on would be medium viscosity, in addition to probably mechanical forces. If we are dealing with a compound that belongs to BCS classes 2 and 4, then we need to give much attention to pH, buffer capacity, and surface tension changes, in addition to micellization. So, because in this case, the disintegration is probably no longer a rate limiting step. I mean, in case of BCS class 1 and 3, with the slowing down of disintegration by viscosity, it would happen that disintegration would be the rate limiting step, and here, so disintegration is affected mainly by what? And the fed conditions, mainly by the viscosity increase and by the pattern of the mechanical forces exerted on the tablet. However, if we go to BCS class 2 and 4 compounds, then we have the issue of the dissolution and accordingly the solubility of the dissolving compound, and so we need to take into account that the pH changes, the surface tension changes, micellization of lipidic components, and buffer capacity changes as well. So first of all, we need to identify on what do we need to focus. So maybe in BCS, so for BCS class 1 and 3, it is a bit simpler, while for BCS class 2 and 4, BCS class 2 and 4, we need to add additional layers of complexity to the viscosity and mechanical force, and we need to have for that we need to have good physiological data for those pH and buffer capacity and interfacial phenomena related parameters.
Thank you so much. Dr. Langguth online, do you have anything to add to your colleagues' comments, or should we go on to the next?
I don't have much to add, just that the dissolution will be even more complicated. We saw that in a previous presentation when a zinc, an additional octanol zinc was added to the dissolution model in order to allow for the uptake or the solubilization of the low soluble compound. So that would just show what Jozef has mentioned, that additional aspects in terms of the dissolution model for low soluble compounds need to be considered.
Thank you. Thank you. Dr. Cristofoletti?
Well, yeah, I think I don't have much to add. Just kind of, I think it really when it comes to what can make an experimental condition biopredictive, I think one element that we need to keep in mind in that is that maybe that is not going to be one solution fits it all. So maybe it will really depend on whether we have a food effect that is affecting the drug itself somehow, or whether this food effect is kind of also being generated due to an impact of the food on the formulation as well. And that may create an additional complexity. So I think it's more like that needs to be considered, whether the food effect is being elicited by an impact of the food on the drug itself or on formulation, or a combination of both.
Thank you. Thank you. Move on to question 3. What knowledge gap do we see, and what research can be done related to oral PBPK modeling to support ICH M13A for evaluating food impact on BE? So first to Dr. Zhang.
Okay, thank you. Yeah, thank you for the invitation. So I'm Lei Zhang. I serve on the M13 both as a rapporteur, and so I have to look at all the aspects, and also as I'm from the FDA. So I'm kind of wearing both hats. So I just want to remind everybody, you know, where we are here today, right? It has, I think even before the ICH, FDA already started research on, you know, studying the food impact on the fat BE, just because FDA is different from most of the other agencies. We require both fasting and fat study to begin with. I think this research is really support the FDA and also help us move forward towards a harmonization. I think we did have a big jump. If you look at the draft M13A, we do make a risk-based decision on whether you need both fasting or fat study, which probably can eliminate or move away a lot of fat BE study, which are currently recommended by the FDA, if the formulations can be classified as non-high risk. But we are not completely, as a guidance, you have to see, just like what Susana also talked about, science probably always move faster than what can be reflected in the regulation. Not to mention, this is the ICH guidance. We have to get agreement from all the regulatory agencies. Some are more experienced, others are less experienced. So I think right now the difficulty is we do have a 2 bucket, right? High risk and low risk. So, and there are probably a lot of products still in the middle. And it's very hard for the M13 to really delineate very specifically what that is. So we can only lay out the high principle. I think we already moved way ahead, like a very, consider the research things like more than like in the last decade. We are confident to say maybe, you know, we are open the door to say you can use PBPK to help the regulator to maybe justify you don't want to do that fat BE. I think that here the risk is mainly related to the BE risk. Like when you only conduct one study, either in the fat or fasting condition, can you extrapolate to the other condition? So I think the mechanistic understanding on the food impact on the drug, on the formulation, and on all these interactions is very important. I think that The gap, I would say, is pretty much, I won't repeat because you heard from this workshop and the other workshop. These are the knowledge gap that will help, but how quickly they can put it into M13, probably that's not going to be that quick, but we can lay out the principle. I think the opportunity could be for the M13 to consider, because we did put that draft there, we got a recommendation or comments. We are reviewing those comments. I think another space is that we may, because we all know there's no kind of harmonized way of doing the modeling, right? That's not there yet. So for the M13, I think the good way is we probably gonna create a question and answer document that may be some space for us to give some example to where maybe we think a low-hanging fruit more confident. By most of the regulations, but some of the area maybe it's less confident. So for the applicant, we always encourage them to keep using those modeling approach and to make the case. Depend on the fit for the purpose. I think you can always go to your target agency to make the case. And also publicize those cases. I think that can move the field forward and make the regulatory agency worldwide to be more confident in use those models for the regulatory purpose. I'm going to turn over to Susana as well. She is also part of the M13, and I believe we also have Paulo online, who is from EMA, can also comment on this question.
Thanks, Lee. So I think you raised a very important point. We're writing a guideline now that will be with us for a long time. So we need to make sure that the wording of this guideline is future-proofed and opening the door as science evolves to incorporate new methodologies. I don't know, Paulo, maybe you want to comment further.
First of all, I would like to compliment all that are in the panel and in the audience. Regarding this topic on the use of PBPK for the M13 bioequivalence guideline. Well, indeed, since we require now in the document that the applicant justifies why he should use fasting and fed studies or just one of those, we give some indications of what we expect the applicants to provide, but we are also opening the door for PBPK models because of that mechanistical explanation that may help the applicants on their decisions. The reason for it to be there is kind of illustrated by the presentations that we had today, where we saw some good examples of what can be done, but also some question marks of what are we certainly sure that the model is giving us, and the confidence that the model is giving us, and where it can be improved. And of course, this line in the guideline goes in that direction. We know that this is happening. We know that this will probably be better in the future. We are dealing with model-informed drug development guidelines. We have also guidelines on the use of PBPK model. We are paving the way for this to happen in the future, although in the present time, maybe it's still not the case. Maybe we are still not using the full potential of these models for regulatory purposes because of the relevance of it, and also the risk-based approach that we are taking notion it. So for my part, I don't know if you need any further view. Thank you.
Thanks. We have one comment for the audience, and yeah, we've got about, I think, a little under 15 minutes left, so we'll try to keep our comments and responses brief. Yeah. Please.
Thank you. I'm JD from Boehringer Ingelheim. Have a point, yeah, let's say provocative thought here, because I don't think that the problem relies really on the fed biorelevant media. I think that fasted solubility is normally underpredictive of the in vivo case. And that's why, I mean, we can do improvements on the fed conditions, but that tells us how high can we go. But the problem may not be there, but it may be that we are too low here and we are artificially thinking that this difference between fasted and fed is the food effect. But maybe the fasted needs to be more relevant of the in vivo case. And then this difference between fasted and fed will be more realistic. What do you think about it?
Does anyone have a comment, or people just want to think it over a little bit and can discuss during lunch? Fang, did you want to?
Yeah,
Okay.
I think the PBPK modeling may help to evaluate the risk if we would like to waive the FAD-BE study and the PBPK model and incorporating the dissolution, because we are right now doing the research to support this. And what would be the dissolution methodology to mimic the in vivo situation under FAD condition? That could help. The fasting and the fat, if there's in vivo data, you try to incorporate the dissolution and predict the in vivo data. That could help us to understand. And also, based on the literature, if you understand the excipient food interaction and how that can impact the reference and test the product differently, that will be also helpful.
Great, thank you, Fang. So let's move on to the next question. Thanks for the question from the audience. What are your experience? What are the experiences you have using PBPK modeling to evaluate food impact on bioequivalence from an industry or regulatory perspective? So we'll go to Dr. Ahmed.
Yeah, thank you. Can you hear me?
Yes, we can hear you.
Yeah. First of all, thank you very much for giving this opportunity. And just to share our experience, I mean, for a couple of IR as well as modified release products, our experience has been pretty mixed in terms of predicting the food effect and interaction with different regulatory bodies. And especially for class 1 and class 2 drugs, it has been, we have seen a certain amount of success as well. But apart from that, we've also seen a lot of challenges. I think much has already been discussed, but just to reiterate, having a fed biopredictive media, and what we have seen is that in terms of simulation, we were more successful in simulating a positive food effect, whereas in cases where there was a negative food effect, there we had a lot of challenges. There was paucity of data, and there were also regulatory questions. And one area was in terms of understanding the mechanism of food interaction. So I think those are some areas where we have seen challenges, but yes, we have had our mixed experience. And I think from an industry perspective, because we are going towards harmonization, and for certain markets, we have already talked, we only need fasting studies, but for FDA, we need both fed and fast. That brings in a lot of burden to the industry. But obviously, if we can justify, we are looking forward with the initiative in M13. We are looking forward if we can, with the entire support or validation activity, we can go to a stage where we have a robust model to justify the fed studies. I think that is what we are looking at. And from an industry perspective, we look forward, and we are there to support in this initiative. Thank you very much.
Thank you. Thank you. Let's go next to Dr. Jianghong Fan, please.
Thank you for the question, and thank you for the invitation. Since I work at the Office of Clinical Pharmacology at FDA, so I will provide some thought from the clinical pharmacology perspective. As a reviewer, if I have a submission in which the sponsor used the PBPK model approach to evaluate the food effect, so as a reviewer, I will check whether the model incorporate the key factor that may drive the food effect. Definitely, it's not really easy to identify the key factor that may contribute to food effect. I think it really depends on how well we understand our drug of interest, how well we understand the drug product, the formulation, and how well we understand the interaction between the drug product and the physiological change and the fat condition. It's not an easy question. So I think once the model incorporates those key factors, the model may be used to evaluate the potential effect of the food on the bioequivalence. Especially, I think we can use the model to do the risk assessment. So for us, I think once come to the final decision, we decide whether the food really have impact on the bioequivalence. I think should really based on the totality of evidence. At this stage, I think PBPK modeling approach may not be considered as the sole determinant in this regard. Thank you.
Thank you. Let's go on to Dr. Kolhatkar.
Hi, so I agree with what Dr. Jianghong mentioned in her response. As we have been discussing today, the agency is open to near approaches which are different than what's typically recommended in the PSG, including use of PBPK modeling to understand impact of food on bioequivalence. I can share some comments and recommendations based on my experience in the regulatory agency. So the parameters that are used to build the model should be appropriately measured or adequately justified. Dissolution data, if they're part of the model, appropriateness of the method should be supported by the model predictions and adequate validations. The agency generally recommends using all available data. So if, say, a waiver is requested for a Fed-BE study, and if fasting BE study is conducted with the test product, then using that fasting BE information is helpful to see if the model has captured key aspects of impact of food. We should keep in mind the purpose of the model as well as the risks involved as the model is being built and validated. And having good validation increases confidence in the model. Similarly, having good understanding of the formulation and how excipients can interact with food is also helpful. And always the final decisions are made based on totality of the provided information.
Thank you. Fang, do you have anything to add to that?
Yeah, I agree with the other members' thought. And then I also would like to comment based on our experiences. We discussed with the industry in some meeting packages on whether the PBPK can be used to support a waiver of the FAT-B study. For that case, we found that the predicted PK is not sensitive to the dissolution rate incorporated into the model. So we suggest one of the comment is that please demonstrate the biodiscriminatability of the PBPK model. And also I would like to suggest if you have any questions regarding the using PBPK model, other modeling for support your B assessment for ANDA submissions, please you can use the MIE pilot program that we just launched.
Thank you. And online, Rebeka, Jereb, do you have anything to add?
Yeah, just some industry experiences from our company. So as presented, first of all, I would just say that we see FED studies less critical than the fasting BA studies. But anyhow, some modeling we've done internally and also discussed with health authorities with regards to these options of waiving FED bioequivalent studies when they are required. So we developed the models on literature data, originator data, and in-house pilot studies and pivotal studies in fasting and fed state. So we were more or less confident, but as I mentioned, we have still some challenges with developing and validation of model, especially validation. So the last thing that Fang emphasized, so that having the discriminating method to really see the bioequivalent and non-bioequivalent batches with your model. So we don't have a successful case, but we've been discussing with health authorities to see if we can find a common path for the future.
Thank you. So just about 4 minutes left and we're up to the last question. So should we use kinetic or thermodynamic solubility to model drug release from amorphous solid dispersions? Dr. Cristofoletti?
Thank you for that. So I would say that for amorphous solid dispersion, it could be a good idea to try to use this apparent kinetic solubility. This is going to give us a better input. We tried both approaches in our study case. And I mean, using the solubility, the thermodynamic solubility for the crystalline form, using thermodynamic solubility, maybe we may have some precipitation over time, but as long as we may have some stability of this supersaturation over a physiologically relevant time span, I think that could be a good alternative to inform the PBPK model.
Thank you. And we're going to go to the audience. And
yeah, I have a question. This is Abdullah from DQMM. So it's more like a comment. Like I feel like we have more chance of improvement or developing something when we have negative results available. Like sometimes we try to validate fed model, but we couldn't. But those examples sometimes are not very available, like successful cases. So I would request, or maybe it's more beneficial for a whole community, the negative data that are also published. It may not be, sometimes journals are also like against publishing negative data, but those things are very helpful to find out where the challenges are, like where the difficulties that people are facing that we need to improve in terms of FAIR studies. So that's something I realized when I was working, like if we have challenging data or negative data that were not successful, if we have the available of those data, those will be helpful for all of us to improve the situation. Thank you.
Thank you. And Andrew, did you? Oh, okay, great, okay. So we'll switch back to the last program question for today for online. So Mohan, Rayeni, did you want to comment on the thermodynamics solubility question?
Yeah, thank you for the question, and I completely agree with Dr. Cristofoletti, and so my experience was also similar to it. And whenever we are testing solid states which are not the most stable ones, and kinetic solubility would be more advantageous for the saturation solubility, because I'll give you an example of a amorphous solid dispersion where the polymer is acidic and the drug is weakly basic. And when you drop it in acidic media, it tends to leach, and when the saturation is reached, it tends to precipitate. And in the physiology, in that condition, both AST as well as the crystalline free base would coexist, and both will have a relatively different solubilities. So especially in such cases, it's better to have both kinetic solubility and correlating the life through which it exists in a solution form to the physiological factors or the PK parameters would be more advantageous, and that being the model input would give us a better predictive models. That is my opinion. Thank you.
Great, thank you. And I think that'll be the last word for today. So we just want to thank the panel, people here today and online, and also thank the audience for some great questions. And also just remind everyone too, for that's in person, there'll be some very interesting roundtable discussions today. So with that, we're gonna wrap it up. And